You must have heard of the four main types of blood group. They are A, B, AB, and O. Wait a second. How did AB come about? And O, why not just type C or type D? Hi, welcome to our first video here on SOS. Today we are going to be discussing about something pretty interesting, and that's antibodies, antigens, blood group, and also a little bit about vaccines. Before we can answer the questions that we started our video with, we need to first understand what are antibodies. Antibodies are protein molecules that are produced by a specific type of white blood cells known as B cells. B cells are also known as B lymphocyte cells, and they are not the ones that goes around and engulf foreign particles to destroy them. Instead, they produce antibodies which contributes greatly to our immunity. Most of you probably will not need to know what is the structure of antibodies, but as the theme of biology goes, understanding the structure will help you appreciate its function. Every antibody molecule is made up of 4 polypeptide chains, namely 2 heavy chains and 2 light chains. The 2 heavy chains will form a Y-shaped backbone, while the 2 light chains will then attach itself to the 2 fillers of the Y-shaped structure. Do note that this fillers term is something that I casually coined onto this structure because it serves a very similar function. Let's take a pause for a moment and think about this. Does having the strongest armory always guarantee that you can defend against an invasion? In fact, it's not as simple as that, right? You need to first be able to detect that invasion. If not, even with the strongest armory, you would not know when to use them. And so that's the role of antibodies right here, which is in the form of detection as well as helping with the defense. Antibodies have a specific ability to detect certain specific structures found on foreign body, and we call these structures antigens. And hence, we can define antigens as a specific structure that triggers an immunity response. Where can we find antigens? Antigens can be found on the surface of foreign bodies, which include bacteria, fungus, viruses, as well as organs that are being transplanted or blood that is being transfused. Now, recall back to the Y-shaped structure of antibodies. Remember the 2 fillers that I have previously mentioned? Those 2 ends are actually antigen specific. This means that a specific type of antibody will then trigger a specific immune response particular to that antigen. And that makes sense, right? You need to have different strategies for different types of invasion. Alright, now let's zoom in into the functions of antibodies. We can broadly categorize the functions of antibodies into 4 main types. Firstly will be neutralization. Secondly will be complement activation. Third will be opsonization. And fourth will be agglutination. Let's start with number 1, neutralization. As you already know, the top 2 ends of our antibodies will attach itself to the antigen. And this itself may result in the inactivation of toxins or just prevent the virus or the bacteria from binding onto its target cells, and hence that render them ineffective. As simple as that, hence neutralization. Number 2, complement activation. You might be wondering, hey, we have always been talking about the top 2 ends of our antibodies. What about the tail end? And that is where complement activation can come in. Okay, to put it simply, the tail end of our antibodies can contain some components that will trigger complementing actions to take place, such as the destruction of a cell wall or cell membrane, or just basically a whole sequence of chain reactions that can be promoted thanks to whatever that is being tagged on by the tail end of our antibodies. Now moving on to the third feature, which is the opsonization. I'm sure you have seen a lot of, not a lot maybe, I'm sure you have seen some, actually I'm not so sure either. You might have seen some movie clips where the protagonist or some high-tech stuff where they aim a laser at the target and then airstrikes start coming down on that target thanks to that laser beam. Opsonization works something like that. What opsonization is about is basically the antibodies tagging these foreign bodies and hence attracting the phagocytes to come over and destroy them. As such, some actually classify opsonization as a type of complement activation. Now moving on to the fourth and the last one, that will be agglutination. And agglutination is very interesting because it brings back the idea of understanding antibody structure. As you can see, there are two ends of an antibody and each end will recognize or each end will bind itself to an antigen. Since there are two ends, if one end is to bind to the antigen found on, let's say, foreign body number 1, the second end is available to be bounded onto the antigen found on foreign body number 2. So thanks to one antibody itself, it can actually attach itself to two separate foreign body. And with that, it may result in the clumping of foreign bodies. Can you see that? Can you imagine that? So with this action, it seems to tie all these foreign bodies together into a clump. And that's basically what it means by agglutination. With agglutination, it not only neutralizes foreign bodies as they are clumped together and limits their movement, it also allows a more efficient phagocytosis to take place. Now that you have a solid understanding of antibodies and antigens, we can answer our initial question on why is there blood group AB and blood group O. You might have already guessed it. A, B represents the type of antigen that is found on our red blood cell. I repeat again, it is antigens and not antibodies. And that makes sense because a blood group AB will then means that both antigen A and antigen B are found on the surface of the red blood cell. That being said, blood group O means that there are no antigens found on the surface of their red blood cell. And that is easy to remember, doesn't it? Because the letter O conveniently just looks like a naked red blood cell. Naked. Now that you have understood that the letters A and B in a blood group represents antigen found on the red blood cell, we can move on to a very important question on whether antibodies A and B should be found in a person's blood plasma. You might have realized that I mentioned the word plasma, blood plasma instead of blood. And that is an important distinction. In order for a person to survive, it makes sense that antibody A should not be present together with antigen A. If not, agglutination will happen. Hence, we can say that for a person who is of blood group A, he or she should not have antibodies A. In order to differentiate between antibodies and antigens, we have a specific form of writing or format of writing, which is to represent antigens with the capital letter and the antibodies with a small letter. So bear that in mind. Now that we have established that the antibody and its corresponding antigen should not be present at the same time, we can then infer that a person of the blood group A, capital letter A, should not contain antibodies A, small letter A. But this person will have antibodies B because antigen B is not present in his or her blood. That also means that the person cannot receive any blood that has antigen B on it. And that means any blood of blood group B or AB. Does that make sense? Because once again, B and AB represents antigen. Aha. So with this understanding, can you then infer which blood group is considered as the universal donor, meaning that they can donate blood to all types of blood group, and which blood group will be considered as the universal recipient, meaning that they can receive blood from all blood types. One important point to take note is that whenever we are talking about blood transfusion, we are usually referring to red blood cell transfusion, meaning that only the red blood cell is being transfused over to the recipient. This also means that the antibodies which are found in the donor's this also means that the antibodies which are found in the donor's blood plasma will not be transferred over to the recipient. The universal donor is none other than... That's poor sound effect. The universal donor is none other than blood group O, because their red blood cell is totally naked. Having no antigens found on the red blood cell also means that it has nothing to be recognized as foreign by the recipient's antibodies, hence making it the most favorable blood type for blood transfusion. Next, you should have guessed it already, so the universal recipient of blood is none other than blood group AB. This is because a person of blood AB should not have antibodies A or antibodies B. And that means that they will not have any immune response to any type of blood, be it O, A, B or AB. With that said, the most ideal type of blood transfusion will always be that of the same blood group, meaning that even though a person with blood AB can receive blood from O, A or B, it will still be most ideal to transfuse blood of the blood type AB itself. The reason being that there may still be traces of antibodies that are not being siphoned out from the separation process while the donor's blood is being processed. Now that everything about blood group is crystal clear, it's time to practice. Head on down to the link to our website and download the very nice diagram to test yourself to make sure that you have understood everything, and then cross-check it with our very nice and succinct diagram with all the answers included. Last but not least, we want to mention something about vaccines. So how does vaccination works? And how is it even related to antibodies? So what exactly happens when we go for vaccination? It's like a mock scenario if the invasion of a certain compound is to happen in your body. Something that contains the antigens that are found in that particular disease-causing pathogen into your body. So the idea is that by increasing the antigen count in your body will respond by producing more antibodies. That's right. So the next time when the real infection were to happen, your body is already armed with a lot of antibodies and ready to counter that infection. That's it from us today. We are sure that you have learned something about antibodies, antigens, blood groups, as well as vaccinations. So if you like this video, don't forget to like it, give us a thumbs up, smash the bell, subscribe, do all those usual stuff, and we will see you in the next video. Cheers!